My name is Anatol Kreitzer, and I'm an investigator here at Gladstone. And I'm with my colleague Talia Lerner to tell you about a discovery we've made that may one day lead to a new treatment for Parkinson's disease. In my lab, we focus on Parkinson's underlying mechanisms so that we can develop new ways to combat the disease. The Parkinson's Disease Foundation estimates that more than 10 million people worldwide suffer from the illness, a chronic and progressive movement disorder in which neurons malfunction and ultimately die. Parkinson's disease is caused by a loss of neurons that produce a chemical dopamine in the brain. Now, standard treatments for Parkinson's for many years have been to replace the dopamine that's lost. This involves drugs such as levodopa, which the brain can then convert into dopamine and replace the missing chemical. Dopamine replacement therapy has a number of problems. As more and more dopamine neurons degenerate, patients must take an ever-increasing dose of the drug to maintain a therapeutic benefit. However, this leads to a number of unwanted side effects. Therefore, it is important to develop new therapies that are independent of dopamine in order to treat these patients with advanced Parkinson's. Parkinson's disease is caused by the loss of dopamine neurons in the basal ganglia. And the major target of these dopamine neurons is a brain region called the striatum, which receives much of the inputs into the basal ganglia. The striatum consists of a number of different cell types, but there are two major projections out of the striatum, and these are called the direct and indirect pathways. And we believe that in Parkinson's disease, there's an imbalance in these pathways, such that the indirect pathway becomes too active and the direct pathway is not active enough. And therefore, we focused our studies on what we term synaptic plasticity, or understanding how the strength of inputs onto these different neurons can change the activity in these circuits. A major form of synaptic plasticity onto direct and indirect pathway medium spiny neurons, which are the major projection neurons from the striatum, is called long-term depression, or LTD. The first step in the study was to identify the signaling pathways that lead to the induction of LTD in indirect pathway medium spiny neurons.
Here in the Kreitzer lab at Gladstone, we use a technique called brain slice electrophysiology to study synaptic plasticity. So the first thing that we do is create these brain slices using a vibratome, which is an instrument that allows us to make very thin slices that are suitable for this technique. And once we have those brain slices prepared, we can go over to an electrophysiology rig to start recording from neurons in those slices. To test the hypothesis that overactivated RGS4 was contributing to Parkinson's disease symptoms, we took mice that had been given a chemical to kill their dopamine neurons, mimicking the effects of Parkinson's disease, and compared mice that had intact RGS4 to mice that had RGS4 knocked out, so the protein was completely gone. And what we observed was that mice lacking RGS4 had much better motor behavior symptoms than the mice with intact RGS4. So RGS4 knockout mice were able to move freely about an open arena and were able to do a task that required motor coordination, which was crossing a balance beam.
Therefore, our working model is that loss of dopamine in Parkinson's disease leads to an overactivation of RGS4, and this in turn leads to an inability to undergo long-term depression at inputs onto indirect pathway medium spiny neurons. This leads to an overactivation of this indirect pathway, causing problems initiating movements. Therefore, it would be useful to develop inhibitors of RGS4 as a way of treating Parkinson's disease symptoms independently of dopamine. We are now working with in-house industry experts to develop such compounds, and hopefully one day such compounds may indeed be able to treat human patients with the disease. Our ultimate goal here at Gladstone is to develop new treatments for those suffering from devastating diseases like Parkinson's disease. We hope to see you again soon, but for now, signing off for the Gladstone Institutes, this is Anatol Kreitzer.